So it's fantastic to engage with our natural history societies, and great to see one that's so active. So very impressed and excited to be here. Thank you for the invitation. So yeah, I was asked to start with about me, and so I thought I should start with me, my happy place, right? This is me outside the laboratory, away from the computer, lying in a field with an eyebright plant, and this makes me happy. This is, as a kid, walking on the south coast in Cornwall on camping holidays, seeing our wild flowers, and I always think that everyone celebrates Rhinanthus, our yellow rackle, as our parasitic plant, but I would think that an eyebright like this, which is feeding on its host plant, is equally as exciting. So these are some eyebrights, and Darwin wrote about eyebrights as well, hearing about Darwin just before. Darwin wrote that some young man should continue studying this interesting group, so I thought I had to take on the chalice. So I'll come back to eyebrights because they are fascinating. But yeah, about me, I did my PhD on begonias. So many of you might sort of turn your nose up as begonias, think about the big double floral monstrosities, dare I say, that you get in the garden centre. But if you ever get the chance to drive through somewhere like Mexico, they are extraordinary. We just hit B2K, which is our big milestone in the begonia world. Our 2000th begonia species described to science. So that's B2K. And during my PhD, I had an amazing time climbing the mountains of Mexico, and you find that on each mountain you get a population that has a slightly different leaf shape, and then when you do the genetics, you find that each mountain, even if they're only a few kilometers away, is incredibly genetically different. So you start to realize that there's so little pollen flow and there's so little seed flow that each population is actually its own species, and they're hard to cross. So that was a fascinating voyage into the world of begonias. And because we're surrounded by natural history enthusiasts, I thought I should say that I also love bird watching and dragonflies. I don't know if anyone recognises this absolutely stunning dragonfly. Does anyone recognise this one? Has anyone been travelling in the States? And if you ever get the chance to go to Florida, don't go to Disney World. Go south, go to the wetlands, and this is called the Halloween Pennant, which I think is such a lovely name. And if you go to the Everglades, you will see clouds of this dragonfly. There are the dominant species there, and I've always been fascinated by dragonflies. The dragonfly fauna of Britain and Ireland has transformed in the last 20 years with climate change, with new species coming in and colonising. So many of the species are completely new to what's I remember as a kid, and it's a fascinating world. So that's me. But today I have to tell you about genetics, and I need to tell you about species identification. So I thought I should start by telling you why identifying species matters. And again, to this audience, it seems very basic, and I almost don't need to say this, but I thought I should. So if you go down to the woods today, you might be surprised. So here are two roughly similar plant species. They're not in flower. You might be searching for your wild garlic to make your lovely wild garlic pesto for your dinner. So you might go and pick a few leaves of the one on the left, a few leaves of the one on the right. But then I should warn you that the one on the right is not what it looks like. Of course, when these flower, your wild garlic is on the left, and this is lily of the valley, incredibly toxic. If you were to consume lots of this, you would be in hospital, and species identification matters. If you get the wrong one, you're in trouble. So, I mean, that's a very simple toy example, but then, of course, there's many examples about where species identification matters for real conservation purposes. So this is aloe vera. I'm sure many of you buy this in sun creams or in lotions. It's of course huge in the cosmetic industry. This species is not conserved. You can plant it, you can grow it, it's very widely cultivated for lots of cosmetic uses. But there's many close relatives, like this lovely Aloe henleniae from Madagascar, which is critically endangered, and yet people are starting to harvest this critically endangered species in the field. So of course, we want to know as consumers that we're buying a sustainable, harvestable plant which is appropriate, and so we need to be able to identify this type of species in the cosmetic industry and root out anyone who's illegally harvesting material. So this type of thing really matters in terms of conservation. And then of course sometimes species identification makes the headlines. I wonder who remembers this scandal, the horse meat scandal. I'm sure some of you remember a few years ago it was a scandal that was found that people buying their frozen lasagnas from Tesco, that when they started to identify the meat in it, alas, it wasn't beef, it was horse. So this was a scandal with your Findus lasagna down here. So knowing your species matters, and there's many good reasons why we need to identify the world around us. But plants in particular, which is my passion, can be incredibly challenging to identify. So I've got 3 examples here. On the far left-hand side, this is someone in a tropical forest. Some of you might know, if you go to the Amazon rainforest, in 1 acre of forest, you can have more plant species than all of Europe. You know, it's remarkably diverse. Hundreds and hundreds of similar-looking tree species. So if you want to do this, it doesn't have flowers, it doesn't have any of the key characteristics, you're in trouble, right? That's a hard identification problem. The middle picture, there's a lot of researchers now who are interested in plant sediments, well, identifying sediments and plant species. So these are different cores where people have dug through into the sediment, and you want to identify 5,000-year-old pollen, plant pollen from these cores. But you'd have to be a real pollen specialist to be able to identify your exact plant species from these. So that's a challenge. And then on the right here, we have a crime scene. And you might be surprised to hear there are cases where plant identification has come into criminal cases. There's examples of some very troubling crimes where there's been a murder or there's been other traumatic incidents where plant material has been found on the victim. And then of course if we could identify that plant material, that might tell us, did this event happen in a particular woodland, or can we identify that house plant? Might that tell us something about the victim or the suspect? So lots of reasons why plant identification is challenging. You might have juveniles, you might have incomplete material or degraded material. So what would we like? Well, I think some people would like this. Some of you might recognise this from Star Trek, either the original or the rebooted one. This is the Star Trek tricoder, and you might remember you can hold this up to any organism, and up on the screen it comes up and tells you what it is and how healthy it is and where it's to be found. So this is for many people, the dream solution. This one you can buy for £10 on eBay, so I guess it's not really very active, but this is what I think some people would like. Of course not all, a lot of people, myself included, value the natural history guides, but I do think there's a place for some more automated identification, and there's some tricky material which this would really help with. So some of you might already know this, but I think this will be new to some of you, that actually this dream solution is in many ways already here. So has anyone seen one of these before, an Oxford nanopore sequencer? Anyone show of hand? No? Seriously? So you can pay, it costs a few £1,000s, it's a thing of beauty, but this is one of the next generation sequencing platforms which you're welcome to come and have a look at afterwards. where you can go into a tropical jungle, grab your plant material, extract the DNA, load it onto one of these, and this produces millions of DNA sequencing reads in real time. So you can get one of these sequencing. Some of the smaller ones can run from your battery on your phone. The larger one, like this one, this is a large one, I should point out. The larger ones will run from your laptop battery, and like I say, in real time it comes up with millions of DNA sequences. The longest DNA ever sequenced was produced with one of these handheld, portable sequencing devices. Right? This is incredible technology. Something that is remarkable that actually we can generate this type of sequencing data. Of course, that's the sales pitch, right? That's what you'll read in the commercials. The less glamorous reality is not quite as perfect as that. So here, this is an example from friends at the University of Bangor, and here they wanted to test these types of portable DNA sequences in the field. So they went to the tops of Snowdon and started collecting these miniature brassica relatives, and they wanted to see whether this DNA sequencer could identify their species in the wild. And to do their field genetics, they had to actually make a full laboratory in the field to make it work. So you start to realise that actually, if you need to extract DNA, you need to grind your sample, you need to heat your sample, you need to prepare the sample. So then you have to bring all this equipment. In this case, they ran it from their car battery, but this is hardly that portable. But it's a step towards it. There are other bits of kit now. This is called the bento lab, like a bento box, with all of your kit in one box. And this can grind and spin and process your sample. So we're getting there. We're getting towards being able to do large-scale sequencing in the field, which I think is exciting. And we can also do this with anyone who hasn't actually had much experience with genetics. So I thought I'd include this slide because this is me with a group of students and researchers in Edinburgh, and I thought it would be exciting to try and train the next generation to do this type of work. And so I gave all of the students some mystery plant samples. So this is a mystery plant sample. I've collected it around the university campus in Edinburgh. And so each student got given one of these samples. One reminder about genetics, just because I should briefly detour. So for those of you who don't think much about genetics, you can see here that in this toy cartoon, plants have a chloroplast genome. So that's where the photosynthesis occurs. That's small and easy to sequence. There's many copies. And then we have the nuclear genome, which are your chromosomes, half inherited from your mother, half inherited from your father in most cases. So in a plant, half comes from the pollen, half comes from the seed. So this class, over 2 weeks, all of them managed to assemble the chloroplast genome. And most of them managed to assemble much of the nuclear DNA. So 2 weeks, so they did all the lab work, all of the computing, all of the analysis, and then on the last day, I asked them to tell me what their plant was from their analysis, and all of them got them right, which is very satisfying. So we managed to train a group of 12 students to go from no knowledge of sequencing to being able to assemble their own genome. So that's sort of the state of play. And that's with the student cohorts. We're also really pushing the genetics now to the limits, I think, in terms of the sequencing. So the biggest project I'm involved with is called the Darwin Tree of Life Project, which we have the ambitious aim of trying to sequence high-quality reference genomes, so complete chromosome-level genomes, the best that we can sequence, for every species in Britain and Ireland. So we're trying to sequence all 70,000 species in Britain and Ireland. So all the birds, all the moths, or the micro-moths, even the smaller grey moths. They look quite similar to me, but everyone tells me they're very important. So we're getting into moths, all of the fish, and of course for me, I'm excited by all of the plants. So we're doing all of our native species and all of our invasive species. And we've just released our 2000th high-quality genome. So this is now a resource for lots of researchers so they can rapidly progress their research. And we've really pushed the limits of the technology. So we've done quite a lot of the tiny. The tiny are really hard. This is David Bell from the Royal Botanic Garden, Edinburgh. And we said to David that we want to have genetically unique individuals. So he had to go to every clump of moss. He's a biologist, an expert in mosses and liverworts. Every single moss, extract an individual, so a single strand of the moss, and we've extracted from that. Because the technology works very poorly if you mix individuals. It's very hard to assemble computationally. So here's some of those individuals, absolutely tiny, tiny samples that he's working with. I thought I should also mention the giants, but this time the giant genomes. This is a real favourite of mine. This is of course mistletoe. I thought it's Christmas time, so I should mention, well, it's December now, yeah? So I think I can call it Christmas time, or the approach to Christmas. So this photo I took in Cambridge. This tree is absolutely dripping in mistletoe. Mistletoe is another parasitic plant, so it's got a modified root structure that grows into the host tree and steals its nutrients. And of course it's celebrated in folklore, but I think most of us think of it for this. So this is Father Christmas with his mistletoe. But the reason why we've been so excited from a biology perspective is that perhaps this is how mistletoe sees the world. Mistletoe has the largest genome, so the most DNA of any species in Britain and Ireland. It's 30 times the size of the human genome. So mistletoe has a 90 gigabase genome. The human genome is 3 gigabases. So mistletoe's expanded to this enormous genome size. So we thought, why not? Let's try and sequence that DNA. And of course, if anyone knows how much the human genome costs, sequencing a 90 gigabase genome, largest genome ever sequenced, largest species, largest genome in our fauna or flora. So gosh, that was a challenge, and yeah, an interesting one, and very happy to talk mistletoe genomics if anyone's interested. I mean, one brief detour, if you will indulge me. We've become very interested in these mega genomes, these absolutely ginormous genomes. We used to call them obese genomes, but apparently that's not so PC to call genomes obese. But these absolute giant genomes, because no one's ever looked at them. And we were wondering if you get an absolute giant genome, is everything large? And so we identified the centromeres, which are the attachment points, the attachment of the spindle, so the middle of the chromosomes, and we identified these centromeres, and amazingly, each of these attachment points is as large as a whole chromosome in a small plant like this Arabidopsis. So I'm telling you this just to highlight that we understand so little about the big genomes. Conifers have big genomes, some monocots like bulbs, like bluebells have giant genomes, but no one ever studies them because they're so complex, and we're just starting to scrape the surface there. So I should come back to my main theme about plant species identification. So you might be sitting there thinking, what's the problem? I told you our students can sequence genomes. We can sequence the big, we can sequence the tiny. So why can't we reliably use DNA to identify species? Well, I think the biggest issue is that at the moment, we don't really know what we're looking for. We just don't have, like we've been hearing about the archives earlier, we don't have a reference library to look up what we're looking for, and it's hard to know what we're going to find. So let me explore that topic. What would we expect in a simplified case study? So here, if we have an evolutionary tree showing the relationship of 4 different species, if you have an ancestral white-flowered species, you might have 2 closely related species. One is red-flowered, one's pink-flowered. And then down here, we've got a dark blue-flowered and a light blue-flowered species. In this case, if we were to sequence the DNA, you might expect something like this. So in this example, you can separate out the species by, in the top case, they both share a C at that first position, and in the bottom case, they both share an A. So that separates these 2 groups, and then within those species pairs, the top one, the red and the pink, they differ by a G and a C, and the bottom one differ by a C and an A. So that's what we might expect. And that's the logic behind DNA barcoding. DNA barcoding, which many of you may have heard of at some point, is like DNA fingerprinting, which came before it. And this is where we sequence a short, standardized sequence of DNA. And the reason why DNA barcoding is so powerful, there are millions of DNA barcodes out there, and a lot of Earth's diversity has been DNA barcoded. The reason why it's so successful is that we then know what to look for. So for example here, now if this is our reference library of our 4 samples, if someone goes into the field and collects a leaf from a sample where we know nothing about it, here's our mystery query sample where we don't know what to expect. If we sequence that same piece of DNA, we might find that's the sequence, and when we look at that against our reference library, you get a perfect match. So this is ideal, this is what we want, and this is when you've barcoded and generated a library to look up your sample against. So that's DNA barcoding, and in animals, it is remarkably effective. So in animals, we use one single genetic locus, one region of DNA. The name is called cytochrome oxidase 1 or CO1. It's in the mitochondrial genome. So that's a discrete piece of DNA separate from the nuclear genome, which has proved highly informative in animals. And in this study of neotropical fish, here's a lovely neon tetra, they looked at 254 species. 99.2% of them had a unique DNA barcode. So that means if you DNA barcode your neotropical fish, you can then look it up and tell the species. In a study of amphibians, over 1,000 amphibians, they showed that almost all species had a unique DNA barcode sequence. And then in some cases, it does work less well. To be transparent with you, in Diptera, so the fly taxonomy is quite messy. There's sort of taxonomic issues, so we don't really know what we're barcoding, and here it starts to fall down. But in most groups, you can sequence one piece of DNA from an animal and look that up in a reference library and identify the species. And there's some really amazing work in this area. In this really fantastic proof of principle, this is work done in Costa Rica, and this group of authors went out collecting skipper butterflies, and they did DNA barcoding for all of the skipper butterflies they could collect, thinking it was one species. Then when they looked at the barcodes, they started to see the species in the north has one barcode, the species in the centre has a different barcode. And when they got the taxonomists to look at it, they started to see that actually that group in the north had a different wing pattern, and the group in the centre had a different wing morphology. So they realised actually the DNA barcoding was telling them about which species is which, and when you look carefully, you can actually find the features that differ. So this is a great example of species discovery with DNA barcoding. Similarly, in this study, in Canada, if you go out with your net, you net large numbers of insects, sequence that big pool that you've collected from your net, that will tell you exactly how many insects you caught, because you can look them up in the barcode library and it will tell you what they are. There are so many good examples I could tell you about. I love this example. This is, to me, remarkable. If you take a single flower in the wild, extract the DNA and DNA barcode that plant, it will tell you all of the insects that have visited that one flower. And that's because, of course, when a pollinator comes, they might shed some skin or drop some hairs or shed their DNA. So if you sequence that bit of plant, it will tell you all the animals that have visited that. So in this case, they visited a flower and they sequenced the flower and it told them all of the pollinators and insects that visited, which to me is amazing. So with animals, you can DNA barcode things like this. You can go up to these eggs, DNA barcode those, and it will tell you the species that they come from. Remarkably, you can sequence water. So take large quantities of water, DNA barcode the water, and it will tell you what swum through the water. So there's examples where people have extracted from seawater, and then there's some DNA from whales in there, and it's told them the whale species that swam through because that whale must have shed some skin, which I think is incredible. You can sequence flowers, and it tells you the pollinators, and I couldn't help but including that one. You can also sequence feces, and it will tell you what the animal's been eating. So there are many examples where DNA barcoding works. A slight detour, I wanted to highlight that actually a lot of this leads to direct applications. So one of the most troubling examples, but one which it really has made a difference, is that some of you might have heard of bird strikes, which is the really troubling case where a plane hits a bird that's flying, and in that case, typically the flight will make an emergency landing, and like here they've come off safely from that bird strike, but of course the animal will have died and is extremely disruptive. So these are cases that we really want to avoid. In this case, they actually collected the blood from the plane and sequenced it to work out the bird species that it hit. And then they've started to change the route of the planes or started to change the height, so that if you notice a migratory period, there's particular geese in that area, you can then avoid flying at that height. So again, a troubling example, but one way it can actually lead to policy change. But yeah, to come back to plants. So in plants, we can't use the same assay. So in animals, we can use this one region of the mitochondrial genome. In plants, the mitochondrial genome recombines, so it's more complex to work with, and the sequence evolves much slower, so it's not diagnostic. So there's fundamental features of plant DNA that makes it very hard to identify from a single DNA region. This paper proposed our standard DNA barcode for land plants, and they suggested that we should use 2 DNA regions, and that may identify 72% of species to the right species. So we've used this a lot, and talking about the value of collections, we spent a number of years DNA barcoding every species in Britain and Ireland, which was a huge undertaking. And this was work with Natasha DeVere from the National Botanic Garden of Wales, Pete Hollingsworth, Michelle Hart and Laura Forrest in Edinburgh. We sequenced the 2 barcodes plus an extra region for 6,000 specimens from nearly 1,500 native species. This was mostly student projects, so undergraduates working hard, working through all these samples, sequencing these regions, mostly from the archives, again highlighting the value of these dried, preserved specimens. And I don't need to go into this too much detail, but on the left-hand side, you've got how reliably we could produce the data. So you see the top row there is called RBCL, one of our regions. and we were able to produce data for, say, 95% of species, and that's because it's very highly conserved and easy to amplify. On the right-hand side, you can see that actually that only identifies 70% of plant species. So it's easy to sequence, but it doesn't really tell you much. As you go down, you get regions that are more variable, but become harder to amplify different species. So the take-home for this part is actually, you can DNA barcode plants. If you want to know if your tea is tea, Camellia sinensis, we can DNA barcode it, and I can tell you if it's Camellia sinensis or something completely different. In the middle, I've got herbal extracts, and there's been various projects to DNA barcode these to see if they are what they say they are. Big reveal, they rarely are. So often what you read on the bottle is not what you get inside the bottle. Sorry to warn you that, but it's a common finding that people fill the bottles with other less expensive plants or easier things to harvest. You can mow the lawn and we can sequence that DNA, and it will tell you if there's orchids there and some other groups of plants. So good at the broad scale. But as soon as we want to know the specific species, it starts to break down. So this is one of my favourite examples of how badly DNA barcoding can do in plants. This is the remarkable example from willows. You all know willows, familiar species growing by the waterside. We more often study them, the montane willows in Scotland. And in this study, they did DNA barcoding, and they found that a single barcode sequence was shared by 53 different species across 3 different subgenera, so completely different groups of willows, and across continents. So you get the same DNA barcode sequence in North America as in Europe, and shared by 53 species. So this is never going to give you accurate species identification. We see the same examples over and over. If you look in oaks, study of 12 species, none of them had a unique barcode. In berberis, very few. This was a photo I took in China last year when looking at the rhododendrons. If any of you get the chance to see the rhododendrons blooming in China, in the mountains in Yunnan, it's a spectacular scene, though barcoding does not work to identify your rhododendrons. So yeah, lots of cases where barcoding does not work in plants. So the question is why, right? And we need to understand that to be able to improve the ways we identify our species identification. So it could be something that we are limited by the data, that's one potential, or it could be something biological about plants that make them hard to identify. So if we're talking about data, maybe we just don't have enough data. Maybe we're not using enough regions of DNA, and maybe if we added in more, we'd get better. And at the bottom, point 2, maybe we're just not sequencing the right bits of DNA, and maybe if we looked at other bits of DNA, like the nuclear genome, we could do better. Those are potentials. In the biology side, you'll probably all know that plants hybridize. If you get different species together, they'll cross-mate, and then they share their DNA. So maybe something about plants hybridizing prevents many of these methods working well. Maybe it's because we have many cases, particularly in Britain and Ireland, of recently evolved species, or maybe it's problems with the underlying taxonomy. So these are the things I need to cover as we think about trying to improve the barcodes. So in terms of data, here's, I think, a step change that we've seen with the willows. So willows, I told you, 53 species share the same DNA barcode. But with colleagues and friends at the Botanic Gardens in Edinburgh, we've now sequenced 982 regions of the DNA. And when you do that, rather than everything having the same sequence, here you can see an evolutionary tree of these species, and each species is coloured by their name. So you can see that all the samples of reticulata are clustered together, or Rita clustered together, etc. You can ignore all the details around the chromosome evolution around the outside. The important take home is that when we sequence hundreds of regions of DNA, we can identify them. So in the willows, more data resolves the problem. So it's not just a biological problem, it's a technological problem in the willows. How about some of the other groups? Well, I finally get to come to my eyebrows, our eyebrows, which are a passion for Chris and myself. These are completely fascinating because they're about as difficult as it gets for genetic analysis. So there's polyploidy, so that means chromosome doubling. So some species have 22, some species have 44 chromosomes. They're parasitic, so they've got a modified root that grows under the ground and attaches to a host and steals nutrients. That means if you give an eyebright a terrible host, it will grow that big. If you give it a good host, it will grow that big. So if you're using the way they look to tell them apart, it just depends on the host. A lot of post-glacial diversity, so species have evolved within Britain Island. They vary with the flower size, they all hybridize, and many of them, like I say, are 2, 3, 4 centimetres. So there's not many characters to start with if you're trying to identify them. And like I say, Darwin wrote about these for their parasitism and their interest. These are photos of our native species, and you can see them all the way around the coast and in highlands and in grasslands, and they're endlessly fascinating. And so there's 21 species, I should say, at the moment. It's currently recognised in Britain and Ireland. And if you do your DNA barcoding, which a visitor, Shumei Wang, and collaborator Galina Kasarova did many years ago, we found that here our diploids are in red, our tetraploids are in blue, and other species are in black. And here, the only thing to take home is rather than a perfect tree where everything is clearly resolved, all of these are just resolving into this one big cone. So DNA barcoding does not tell you anything about species identification here. So that's based on 2 regions. If you try a different region, the nuclear ITS, you get this. So you get a cluster of tetraploids at the top, a cluster of diploids at the bottom, but no differences between species. So nothing here is really resolving them. So we thought we could do better, and once again, drawing on our fantastic natural history community in the UK, we asked our recorders to send in samples, and this was work with Edgar Wong, who was a project student in my research group, and amazingly 276 recorders across Britain and Ireland sent in samples, and then PhD student Ding spent lots of time extracting 800 samples and sequenced 20,000 SNPs. A SNP is a single nucleotide, so an A, T, G or C, so 20,000 sites across the genome. So what did we find? Can we identify them if we sequence 20,000 short bits of DNA? Well, Ding found that the biggest divide, the clearest result, was something geographic. So she found in the north of Scotland, particularly Shetland and Orkney, you get a cluster shown here in yellow, and then throughout the rest of Britain and Ireland, you get a cluster here in purple. If anyone here is interested in genetics, this is how we often show this. This is called a structure plot. Here you've got each individual shown along the x-axis as a row, and you've got your yellow samples, which are the northern samples, and then everything else is purple. And interestingly, those samples which are different are this species, Euphrasia micrantha. It's one of our most distinct species in the flora. It grows in heathland, it's purple flowered, it's got unique ecology and morphology. So right, we've got genetic distinction of micrantha in the north. How about everything else? Well, we had a complete lack of genetic splits in the rest of our tetraploid species. So this map shows the genetic relationships of our populations, and this is all of our tetraploids, And we thought we might see some separation of species, but instead, all we see is those in the southwest are genetically related, even though there's many different species here. In orange, you've got this English and Welsh cluster, many different species here, but they're all clustering together. So we were really struggling to separate out the species. So we thought we'd try something else. We'd try and sequence the whole genome, and we would do that in a place where we get fewer species. So we looked, I guess this was partly an excuse, but partly exciting biology. We took a trip to Fair Isle. So for those of you who don't know, Fair Isle is the most remote inhabited island in Britain and Ireland. 70 or so inhabitants, and we thought we could study them in a unique, separate island environment. In the depths of winter, seeing Fair Isle in the sunshine makes me happy. It's one of those places to get to where you go on a small boat, and then you go on a smaller boat, and then you go on a small plane, and then you get stranded there for a week because you can't get back. But it's one of those remarkable places that if you ever get the chance to go, halfway between Orkney and mainland Shetland, and this is the island itself, and you get eyebrights all the way around on the cliffs and on the grassland and the moorland, and it's a remarkable place, famous for the bird observatory, of course, where every evening they do the bird sightings where you can hear them reporting what exciting species are coming through. So Chris, myself and others went there, including a very talented researcher, Hannah Becker, and Hannah sequenced the genome of Euphrasia faulensis, our coastal species, Euphrasia arctica, our grassland species, Euphrasia micrantha, and once again did a structure plot type analysis here, and once again could not separate arctica, which is A, from faulensis, F. We could separate micrantha, so here, once again, we couldn't separate the species even with whole genomes. So this leads us to these possibilities that maybe these species are closely related and hybridizing. Maybe there are what we call genomic islands, bits of DNA that tell the species apart. Maybe there are, we haven't found yet. Maybe the species are just not well defined, and maybe it's too much of a mess and we need to change the taxonomy. But like so many groups, sometimes even with genetics, it's really hard to tell species apart. And excuse me for this, but I couldn't help myself. Like in these case studies, it's just impossible to tell the two species apart. In so many different contexts, they are identical, and I'm sure even with genomes, you would be able to separate them. Okay, so to finish up, I've told you today that genomics is increasingly accessible and data can be generated for nearly any species. Our biggest challenge is that we don't have a reference library of life. DNA barcode is useful to identify species groups in plants, and we're getting closer to exact species matches, and there's a lot happening. It's as exciting time as any time to be working in genetics and evolution. There's as much going on as ever, so there's a lot of positivity and excitement to see how the field develops. So I'd like to thank my research group here on the left, who are having a coffee break between analysing their plant genomes, and friends and collaborators and funders. So yeah, thank you.